Yeah, yeah. Oh, great. Well done, Clementine. That's worked.
Those are
Yeah.
the tubulin binders, right? The
Well, we if this is the benzoxazoles, then when we don't really know.
interesting.
That
Okay, all
was the
good.
hypothesis for the benzimidazoles, but not for the, yeah.
Yeah.
But maybe, well, Matt, you're chairing, so
Yeah. Okay. All right. So welcome everybody. Nice to see you all. Thanks for doing the recording, Clementine. That's great. So yeah, this is our October MycetOS meeting. Wendy, thanks for putting the agenda on the GitHub page. That's great. I guess the first thing I was going to mention is that, as per the agenda, the Fanaramol paper is now finished and online. So I just wanted to do a communal self-congratulatory point about that. So really nice paper. Well done, everybody. I think it's a really good job. Yeah. I mean, it's been a long time coming. It's a lot of work over a long period of time. So it's really good to see it out. I mean, as far as I'm aware, that closes that series, right? In the sense that we've got no plans to continue with that set. And we've had requests every now and again for cross screening of those compounds against other things. But that will be difficult because the compounds are in diverse, weird places. So I'm not quite sure we can do much more. Louisa, you've got your hand up.
Yeah, no, sorry, I didn't mean to interrupt my questions just regarding that because I was just updating our OSN page at the NDI, and the phenarimols is the one, the compound, the series that it's on the page that will be advertised for the mycetos and everything. And then my question was to if it was published, if there was any like, I don't know, ideas to continue that, or if not, I would just move it to previous projects, and then I'll be happy to add a new one onto the web page, just
Right.
maybe a different series that you think there's need support, or like I don't know, anything else, then I can add on the web page as well.
Okay. That's great to know. Thank you. Yeah.
Yeah.
I mean, I think there isn't a lot of interest in continuing with that series. I mean, in part because of the mechanism of action, that it's just the same again of the SIP. So I don't think there's a lot of point in doing that. But we should decide between us soon which of our current series would be best to feature on the D&DI page. Yeah. And that depends a little bit about what we talk about today, I suppose. Okay. I mean, obviously the compounds are still there, and obviously people know that it's a live project. So if anyone does from another group, against another pathogen, really want some samples of molecules to cross-screen against something that they're interested in, then of course we can provide those, but it's not an active chemical series anymore. Okay. For the next agenda item was the second series 2, the aminothiazoles. So there's a bunch of different things going on here. So I think that there were samples incoming from Clementine, and Peter was synthesizing some last time we spoke. I guess from the relevant page, which is issue 36. You know what? I'm just going to dump that in the chat so people have got it. From that page there, I guess the question is which samples are incoming from what's on that page, because I think again that would be the end of the synthesis for this series before we try and publish it. So I guess I just wanted to catch up on what's coming in and what's left.
Yeah, from UCID, we just have two, maybe three compounds because we had a student who was doing like a 6-week research project with us. We haven't actually checked the purity of them yet, but I'm hoping that they'll be okay. And I was planning to send them with the batch of Series 7 ones that we made, like this week or next week.
Okay, cool, great. Okay.
yeah. And
Yeah,
I think Dunya,
yeah, carry on.
yeah, Dunya, I think we received some from Andrea yesterday. I think they were also series 2.
Yes, they were also series 2. I think there were 10 compounds, 11 compounds to be tested.
From this series, really? Okay, I
Yeah.
didn't know that. I haven't seen the structures of those, I don't think, unless I missed it.
Oh, Wendy, are you referring to the ones we sent? Because I also sent a few compounds, but I thought they should be arriving tonight.
Yeah, we yesterday we received compounds from your site, so they
From
were
our
early.
side, okay. Yeah, okay, very good. I will attach on the chat the structure. So they're mostly the amides version, the aminothiazolamide version. So I will upload the file now on the chat so you can all see.
Could you please put that in the link that I just put in the chat and Zoom?
Yes, yeah, I'll put it there.
Because
Okay.
we've got to try and make sure we're on top of all these structures coming. So
Yeah.
that UCL has sent some, and Andrea's sent some, and Clementine's about to send some. So we just got to make sure we've got them all in the same place.
Yeah. I
I
will do
know
that now.
the ones that Eric is talking about, they're in the master list already. And
Yeah.
yeah, I haven't given ours numbers yet, but they're in that issue that you've linked to, Matt.
Cool, great. And Peter, am I imagining things that you were sending some of this series, or am I confusing that with
Yep, so
series?
we're currently making them at the moment. We've got from that link that you put in, we've got all the methyl versions that were left on that list, and then we're making the ethyl versions when they get back to the lab after reading week, and then we'll have those alkylated ones. I don't think we'll get the bottom version with that benzene next to the nitrogen, but I think the rest we should have
Okay.
in the
Again, could
next
you
few
dump
weeks.
those? Could you just dump those structures in a picture or something in that issue so
Yeah.
we know that they're incoming?
Yeah.
And again, you're talking about weeks rather than months, is it?
Yeah, it'll certainly be. So they're off this week, and then they're back in next week. So hopefully in the next few weeks we'll be posting.
Okay. All right. So based, because Clementine, you've got the best sense of the series. Based on those structures that are incoming, if we got in vitro data on those compounds, would that be the time to pause this and finish and write up?
Yeah, I mean, I think we're all kind of in the same boat, which is that we were making a couple more of these compounds, not so much because it was crucial for publishing, but because it was easy to slot them into syllabus and so forth.
Yeah.
And yeah, I mean, I have a draft of a paper pretty much ready to go, although I'm sure if I look at it again, I'll want to change a bunch of things before I share it. But yeah, I think once these have been tested, it shouldn't be too much longer until we can get that out.
Okay. That's awesome. Okay. All right, great. Anything anyone wants to add about this series 2? I think we've captured everything.
Yeah, for us it would help if we have indeed a little bit of a planning, because if they come, well, just week after week, then we just wait with the first ones to screen all of them in one go.
Yeah. Yep. And do for this series, do you include a previously known active from the series as a sort of internal control? Because batch to batch variation. I mean, do you ever, for things like this where we screen them over a long period of time, is that necessary, or do you include internal controls anyway so that you're not worried?
We always include internal controls, so we always include no antifungal, known antifungals, but we can also add one of the previous screened compounds if you would like that. That's not a problem, but we always add known antifungal agents where we also know how stable they are.
Right. Yeah. So, I mean, yeah, Clementine, just maybe give that a bit of thought. If you think that there's a positive, like a standard that's worked really well that we've already sent that we should include as an internal check, then we could do that. But if you don't think it's important, then we don't need to do that.
And I think there's plenty of samples on hand anyway that we could use, but the way it's set up already, like I always get confused which way around they are, but the known antifungal itraconazole is working, as our control.
Okay, fine. then we'll keep it like that. All right, great. But yes, duly noted, and maybe, well, it depends, I guess, when the last compounds come in. But if people could keep Wendy updated, that would be very helpful, so that she knows what's coming. All right. So series 5, the benzimidazoles, benzoxazoles series. Again, the relevant issue for this, I will put in the chat so people can see where we're up to. So the new and final data came in from Dunia. Thank you so much for testing those compounds. That's absolutely awesome. The latest versions of the paper file and the SI were indeed on that issue. I've now downloaded them and just had a look at them. So I've got all the latest files now on my computer, but I'll make sure I put them back up there. The latest version of the SI, for some reason, with the addition of 10 more compounds, has leapt from 3 megabytes to 127 megabytes. I'm not quite sure why that is. I assume there's one of those pesky little diagrams has something in it that is a problem. But well, yeah, I'm not quite sure what to do about that. But I think you can still compress it down. So I think it's just a glitch or something. But essentially, yeah, there are 10 new compounds. These are MycetOS 800 to 809. And Andrea has inserted all of the chemical synthesis data into the SI, which is great. There were 4 pretty good compounds there, 801, 802, 803, and 807, which is really useful. And I will add those to the big SAR diagram. They also need to be added to this supplementary sheet number 1, which we've got, which is all of the data for all of the compounds. And my question was that I need a little bit of guidance about which numbers to put in there. Because let me just quickly share something. Sorry to get into the weeds here, but I just need to figure this out. So it's Excel. Yeah, let me see.
Well, maybe I can help you there, Matt, because I took a look at the manuscript too. And what we noted is that Dunia screened them against MF55, but we still didn't screen it against falciform spora or the other strains of Madurella mycetomatis. So therefore you lack some numbers. So based on the discussion we're going to have here, if we include those compounds, yes or no, we need a little bit more time to generate that data. In my opinion, I think it's only in vitro work. Let's do that. Because otherwise those 10 compounds will not be published in any manuscript. Personally, I hope that they are not active against falciform spora senegalensis, because that will not change anything on the flow of the manuscript. Of course, when we find one of the compounds which is active against both, and by the rule set in this manuscript should also have been screened in vivo, then we have a little bit more time to wait, because well, this is the last week of the larvae season, so we're not be able to test this till the spring. But we could also say, okay, at that point we make the decision if we continue with those 10 yes or no in the manuscript. But for now, I would say yes, please let add them in, because they were made and they are there. But that's my opinion.
Yeah, no, I mean, it feels to me like we should definitely include the compounds. And if we can generate the other in vitro data, then that obviously helps in terms of consistency of the diagrams and the tables and the data set. I would say it's one of these cases where you finish the paper and submit it without the in vivo data for the remaining compounds. And if you give a reason why that is, and if the referees ask for it, we can try and insert it when it's available. But we would, my view is you get it into the refereeing pipeline without that, because it's not going to change the conclusions of the paper very much, I don't think.
I think it takes 2 more months to generate the remaining strain data.
Okay. All right. Fine. We can do this offline if you want. The data in the supplementary sheet looks a bit like this, that you're familiar with the structures and all of the corresponding data. And I can send that over. The data that you sent over for the other compounds, it has a sort of subset of that. And so I just want to make sure that I know how to add the new data to the existing spreadsheet, but maybe I can just send you the existing spreadsheet and you could perhaps add those in. So the data all looks the same. I mean, you will require the latest data, you're right. And then
Duya and I already discussed this, and she forgot to put the data in the real database. So it is there, but that's why it's structured differently. So we will put it in the big database and then extract it into the main figure. Sorry, in the supplementary file.
All right, wonderful. Yeah. So the huge SAR diagram for this paper, these are the black ones here that we can easily insert, and it makes a lot of sense, and we can make comments about the data. But essentially, there are two numbers here, and the scheme is down here, so it's the percent growth at 25 for each, and then the colors are the percent growth at 25. So it's those again, it's those, you know, which numbers I include from those, but it'll become obvious from the supplementary table when you've completed it. There's a separate diagram for the MIC stuff. Let me just remind everybody of that too, if I can. 1 second. I'll share. Yeah, here it is. So this is from the paper itself. So yeah, I mean, we have the MIC values, but yes, we don't, I think, yeah, we don't have this, right, for senegalensis.
That's correct. So the senegalensis we will do first, and then the remaining strains, just for consistency.
Yeah. Okay, all good. So yeah, that's fantastic. You're already on top of that. And then oh yes, and I guess at the same time you have to add these as dot points on the other figures, right, for the screening figures, which is fiddly and annoying, but it has to happen, doesn't it? Yes.
It's not that difficult because it's just copy paste the new data in the underlying graphs.
Right.
So
okay.
it will do it automatically.
Yeah. Okay. Two more quick questions. One was that in the paper we have a Hurg calculation from D&DI from you, Louisa. We have a calculation about Hurg, and we just need some comments for the supporting information about how those numbers were generated.
I think it was just probably Stardrop, right? So I can give you that later if I can find a paper or something like that.
Great. Okay, perfect. Thank you. And there was a question about other people to make sure the author list was complete here. And I just need to flag this up in case people need to be in touch with other people about things. So let me just quickly share this again. Oh, it's amazing dealing with this on a little tiny laptop screen, but it's all happening. All right, so here are the authors as it currently stands. We need to make sure that we've included everyone relevant, and if we're not including people, to make sure that we have thanked them in the acknowledgements. So I just need to make sure that if there's anyone here who feels they or someone else needs to be added to the author list, that they ping that to us. We already have a really extraordinary Excel sheet in the supporting information of this paper, which lists all the students who've helped make compounds throughout. So as part of Andrea's and Charlotte's teams. So that's great. But we just need to make sure that the author list as shown is complete. So please let me know if there's any omissions.
I will do, Matt, about that. I'll just send an email to Ben, see if he wants to be included. And
thanks.
And then I guess I don't know, I feel like we could, I was not involved in the draft, but I feel that maybe in the introduction there could be a line about since all of these were done within the OSN, and also it's Andrea has already published some of these in the JChemEd paper.
Yeah.
That could also be a thing a paper to cite, and yeah.
Yeah, so yeah, actually I did ask Andrea about that, because there are a question about novelty here, which I always like to try and put in MedChem papers about how many of these compounds are actually new to the universe, right?
Yeah.
versus how many have been made previously in OSN.
Yeah.
And we don't have a
I
number.
don't know that. Yeah.
Yeah.
Me
So
neither. Yeah, I don't know.
I was hoping to include that in like, well, I mean, the abstract or the
Yeah.
letter to the journal saying X percent of these molecules are completely
Yeah.
new, X percent have been included previously, you know. But I don't know how we get that. So if we had we've got a spreadsheet of all the molecules, I don't know if it's possible to check how many of them have previously appeared in the OSN project. Is that possible?
I can check. Yeah, yeah.
Yeah,
If you just
if I
directly to the spreadsheet, yeah, I can check.
Yeah, if I recall correctly, the 800 the compounds in the 800 number, they were specifically made for this one. The rest was repurposed, but I'm not 100% sure, but that's what I recall from the discussions.
Okay.
Mhm.
It's not a problem either way. It's just nice to be clear about it.
Yeah.
And to some extent, we for quite a few of these compounds, we're including the full experimental for the chemistry. And in a sense, if those molecules are already published, we wouldn't need to do that. But in this case, is it that we've got molecules that were made for the OSN project which haven't been published before?
I think I'm not sure if all of them were published because I think this paper is 3-4 years ago. But Andrea published a JChemEd paper with the benzoxazole series. Of course, it was focused on the educational part of it, so I'm not sure if she included all the experimental part in there.
Right.
But yeah.
Yeah, because I mean, when you include a compound in this paper, you're with all of the NMR and purity data, you're kind of implying that this is the first time it's been reported. Otherwise, you would refer back to the previous. So I just don't want to mislead anybody that's saying that, well, we've got like 54 new compounds, and actually they're not.
Yeah.
And I mean, the chemical detail in this paper is quite small, partly because of that, and so the emphasis is much more on what they're doing and any SAR that we can observe, rather than the chemical novelty. Which again, it's fine. You just got to be clear about it. All right, we'll see if we can get some clarity on that by email.
Yeah, she did. I just seeing here the supporting information. There is NMR, HPLC, HRMS. For some of the compounds, you just need maybe to cross-check if there's a
because we don't really need to include that if it's been published before. It's kind of unnecessary detail.
Yeah.
Okie doke. That was everything I was going to say about this paper. So I think we definitely want to include the compounds, and yes, it's going to involve a delay to submission, but we can be getting the whole thing ready in advance of that, and it's not going to fundamentally change the paper. We don't. I forget what we said about the potential mechanism of action of these compounds. I think we're just making some general remarks in the paper, if memory serves, which makes some sense. I haven't checked that bit for a while, but we're not doing anything extensive about saying what these compounds are doing, I don't think.
Yeah, I think you're right. We mentioned that benzimidazoles had a certain mode of action, but only that these compounds look structurally similar, and that's why we tested them.
Yeah.
And not hint too much about potential mode of action.
Right. Yeah. I mean, of course, if these compounds did in the end display a novel or less explored mechanism of action, then it might make the series interesting and important again, which is the advantage in figuring it out. But it's not trivial to do that. It's one of those unknowns. All right. The next series was Series 7, which there's been a lot of stuff on the MycetOS website about, and I'm a little bit behind the times here about what's been going on with this series. So is anyone happy to try and give a sort of bird's eye view of what's been going on with this series of compounds?
I know a little bit about it. I mean, Bernard was the one who first introduced it to me, but I went down a rabbit hole of looking into these things. So these are what are called salicylanilides. They're derivatives of a compound called niclosamide, which has been used in livestock and people, relatively safe, but in terms of they seem to, yeah. So there was a couple compounds in in some of the MMV boxes that were derivatives of niclosamide that turned out to be pretty potent. And I think right now it's a matter of synthesis because there's like some safety issues with these compounds in the sense that a lot of the reagents and the salicylanilides themselves are sort of notorious skin sensitizers. So there's a little bit of kind of back and forth. I was talking with Shane about this. Um, they seem to that a lot of the reports seem to say that the amide coupling reagents used to make them and the salicylanilides themselves are skin sensitizers, but they seem to be relatively potent. I think it's just a matter of finding a safe way to make them.
And
Yep.
sorry, I'll just jump in and say I don't think that they're not hard to make. It's just that because we're making them with first years, we're much more limited. And because Shane is our first year lab director, so he is quite keen to do whatever, like he's not shy about trying different methods, whatever else for students. But it's just been the balance of things that you can do with students that also don't cost a ton of money because peptide coupling reagents are more expensive than you would like to use for 60 students at a time and so on. But if a researcher were making these, I don't think it would be that much of an issue.
Right. Right. Yeah. So so for the chemistry, yeah. So Shane, you're doing an amazing job here of doing the open lab book thing on on that issue there. It's awesome. So is it the case that the students who are working on these at the moment at Sydney are using a lab notebook somewhere to record what they're doing? Is that right? Because you can just link to that and then we can see what's going on in terms of the detail and NMRs and stuff like that. Is that what they're doing?
Yeah, definitely. I'm happy to share. I can probably even share now if you want an open view of our ELN. I've shared some sort of random snippets on the GitHub. But yeah, they're not the ones that we made this year, I guess, are not purely salicylamides. We have got the methoxy or the methylated version, I guess, of that phenol functional group. And that was just mainly, I guess there wasn't enough time to sort of roll it out. And I just wanted to make sure that that phenol group was capped to sort of ensure that the chemistry was going to be as simple as possible. And that, that we could work out if there was failure, that it was the chemistry, not the chemist, particularly when you're dealing with first years sort of thing. So now that we know that the chemistry works, then we can start to extend those and start to look at the actual pure salicylamides and possibly try some of those conditions with those to see if they work in the presence of a free phenol.
Right. And so, yeah, again, the sort of bird's eye view of where new compounds. Well, let me just share the SAR diagram because it's a
Yeah, I believe that the phenolic hydroxy is needed. I'm not entirely sure, but as far as I can tell, when you kind of put a bunch of I did some very simple, like using free software electron density calculations, and it seems like you can't you need to have the sort of the chloro and the phenol kind of in that position. And then you kind of need like something that's kind of slightly electron deficient on the other side, on the amine derived side. But you can kind of see that in the SAR, like if you kind of, yeah.
So there's a
like
cluster here of
yeah.
green, right? Which are these? Forgive me for we had a contribution of compounds from Dr. Melendez. I forget the name.
No,
Sorry.
these were Bernard's compounds.
Oh, sorry.
Fluorinated ones. The SF5 one was Bernard's, right? You made that SF5 one, right? Yeah,
Yes,
that one I
this
think.
SF5 and trifluoromethysulfonyl compounds, and yes, these are already published. It's already
Yeah.
published compounds, and I think the other compounds are from the Melendez group, I think.
Okay. So
Yes, these
from
halogenated compounds are from the Werner group, not Retain.
right,
I
right.
think Dunja has tested has some more test results now. Maybe she can tell us something about it
Okay, sorry,
for some
I'm jumping
use.
the gun. If there's new data, we
Uh,
can do that.
Yeah, the issue is you see that methoxy compound there. That is that is one of our hits, I believe, but with the phenolic capped, and it seems no, the other side, slightly more towards the left.
Oh.
Yeah, if you scroll, you see that one, that imid, and then you can see there's a matched pair there where if you cap that hydroxy, potency just dies.
All right.
Um,
Okay, so some of the, but Bernard, some of these are already published, and so we don't want to duplicate that. Are there, promising green compounds in this series that are not yet published that have since been found?
Yeah, that's a good question because I don't know if Dr. Melander has already published some of these compounds before for a different project about new antibiotics or adjuvant antibiotics, and can be the case that they were also published before the chemistry and so on, but they are new in terms of anti-mycetoma activity.
Yeah, okay.
yeah, I
But
don't
Shane,
think.
the kind of compounds you're making, Shane, are they exploiting the greens, or is is exploring something else based on availability of materials?
Yeah, definitely. So the one you have your cursor on there, that MycetOS 8, sorry, the one above, 456. So we basically made the methylated version of that one.
You mean
And
on the oxygen, you mean?
yes, yeah. So
Right.
we've got the relative one. So whether we get the same trend we see for what we saw for the I guess the two trifluoromethyl substitute.
Right.
So I think there are four compounds that we sort of have adjusted. So that was that MyOS 456. There's MMV 665807. Yep. So we made sort of the methoxy version of that one.
Hmm.
MDM 475.
Got it somewhere?
Apologies.
Can't remember where I saw it. It was maybe it's at the top.
Yeah, I think the NDM ones are mostly at the top. But as a side note, I couldn't find the MyOS codes or the testing data for those MMV ones. And I'm assuming, well, I know we've tested them, but I don't know if I just couldn't find them because they're in one of the different tabs. But they don't seem to be in the master list at the moment. And I had to look in the other tabs and I couldn't figure out what was going on.
If they are an MMV compound, then most likely they are in the MMV boxes tab.
Yeah, so when I was looking through those, like Control F, and they weren't coming up. So I don't know. Yeah.
There's a publication of one of my other PhD students, and she also looked at an MMV compound. Maybe it's in that publication because it was not screened in the MycetOS. She screened it at a different university.
Oh yeah, what?
Yes, that MMV compound, I think, was part of your first
Yes.
publication about nicosamide compounds together with a
Yes.
group in Switzerland. I think Bresser.
Yeah.
It's sort Switzerland, based in
There was
Switzerland.
okay. On the front of
should be-
Series 7 in the title page, there was a compound that I didn't know the name of, so I kind of erased the name. It's an isomer of that imid compound. It's down below. It's a positional isomer. That I think had an MMV number on it, but I don't remember its name and I don't have access to ChemDraw. I personally just called that compound imid positional isomer because that's what it is, but
And then yeah, of course we also made we attempted to make the methylated version of the nicosamide. Unfortunately, that was the only one that failed to go.
Also have some just kind of proposed new ideas. There's a bioisosteer paper. We could possibly maybe make something from chlorosalicylic acid and amantadine, which is already available as a very cheap pharmaceutical. For example, to see if we can kind of replace that trifluoromethylanilene portion with something, and I don't, kind of escape from flatland.
Yeah, so my first question, which maybe Wendy or Bernard would already know, is for the actives, by which I mean the greens here, do we know or have we seen generic cytotoxicity? Is that known? A very simple compound.
I think they were non-toxic to the larvae, as far as I recall our paper. Yes, I think they were non-toxic, but their activity against the infected or against the fungi was not so very much expressed. So there's some space for improvement in vivo activity. And I think that IMD compound was a little bit more active in vivo, non-toxic either. And maybe that's a better way to design a better strategy to design new compounds to focus on that IMD compound.
Yeah, that's what I thought of with the bioisosteer replacements. I thought maybe we could replace that bis trifluoromethyl benzene with something like adamantane or cubane or something. Unfortunately, a lot of these reagents are horrendously expensive. I looked at the price of cubilamine. It's like $1,000 for 500 milligrams.
So the IMD compound that you're talking about which
one you have your mouse cursor on, the one with the
This
two
guy?
the trifluoromethyl one,
Right, this guy.
trifluoromethylaniline derivative. Yeah,
Okay.
this
Right.
trifluoromethylaniline.
And again, I haven't done the reading, right? But is it known that the molecules of that type are not toxic? Are they used for anything else? Have they got any other kind of bioactivity that we could call upon?
Niclosamide is used to treat tapeworms, but it's not very well absorbed from the gut. That MMV compound, I don't know what it was used for. These compounds have been used in the past as agricultural fungicides. I don't know why they fell out of favor, but they were used in the past. There were a couple compounds. There's one called benodinil, which is literally where it's salicylanilide, the parent compound, but with the hydroxy replaced with an iodine. And these are known to act on a mitochondrial enzyme called succinate dehydrogenase.
Mhm.
It's also an I kappa beta inhibitor, part of the NF kappa beta pathway, which we stumbled upon in mass research.
Right.
These compounds are very likely multi-targeted. They're pretty non-toxic for being so aggressive. Let's just
Yeah. Any suggested mechanism of action, or is it just I mean, for in terms of the parasite?
I
mean,
would have to.
yeah.
Sorry. I guess I would have to have someone do like docking studies, but maybe succinate dehydrogenase.
No, I wouldn't worry about docking studies. I mean, just if there's anything known, is all.
Yeah, so the niclosamide, which is also a salicin, whatever that name is, does target the MGE1 and mitochondrial protein import complex in the tapeworm and parasites, but it also has then an antifungal effect.
That's an experimental observation rather than a prediction, is it?
Yeah, it's from a paper.
Yeah. I mean, yeah. If you try and dock this into candidate proteins, it'll probably dock into most things, I suspect. So, yeah, you want a kind of experimental observation of some kind of interaction with a target if possible. Again, not a problem. It's just they are remarkably simple molecules. So you imagine they're going to do a bunch of things.
Yeah, I reckon these things probably are pretty multi-targeted. As I said, the toxicity is surprisingly not like terrible for something like this.
Mm. And we may have covered this before, apologies, but for those people who've handled these molecules, are any solubility problems observed in aqueous media? Or are they
I haven't dissolved it or diluted it in PBS yet. I only did in vitro so far in DMSO. But I can look at that as well, because I also need to do the MICs.
right? Okay. Yeah, yeah.
As part of our characterization, we run HPLC, and we also do lipophilic estimation. So we run a bunch of lipophilic standards and then compare their retention time to those. And I think from what we calculated, they're a log D of around about 3 to 4.
Mm-hmm. You don't happen to make, like, 500 milligrams of any of these, like you don't happen to do any on a big scale, do you?
No, I mean combined, I'd have to see how much we've got left. Probably not 500 milligrams.
Mm.
I reckon we might have 500 milligrams of crude stuff, but not of the pure ones.
Right. Because I mean, there's loads of different ways of measuring solubility, and it's possible
Yeah.
that on very small amounts of material. The simplest way of doing it is to take a sample and make a saturated solution, then filter off what doesn't dissolve, and then you've got your number. But you need a decent amount for that to be feasible.
Yeah, I was kind of looking into a little bit of this with the possibility of using bioisosteres for the right hand ring, because yeah, that CF3 phenyl, it's gonna probably pi stack in the crystal structure. I can't imagine IMID being very soluble.
Yeah. I mean, yeah, certainly one way of approximating it is to measure the melting point, because that's a pretty good proxy for solubility. But either way, that you're expecting some issues here. So if you're measuring potency at 25 micromolar or something in an aqueous solution, you've got to know that the molecule is soluble at that level, and we don't really. That's all.
I can go look around for IMID solubility data, because that compound has been used in vitro cancer research for its NF-kappa B inhibition.
Okay, that should be good. Yeah, I mean, it's be an experimental value, right? We don't know any predictions.
Yeah, I'll have to go look around and see if I can find it, because there's got to be something. Because these compounds have been known since, I believe, the 1960s.
No, I'm sure. Yeah, I mean, that is an advantage in some ways. But it would be nice to know, just to make sure that we can put a reality check on some of the assays run at certain
Yeah,
concentrations.
I'll go look around for that, because there's also a compound called trichlorosalicylanolide as well. So that one's also been known since the 60s. So I'll have to go look around, I guess, literature search, see if we can find any solubility data on these things.
Okay, great. Thanks. Louisa, yeah.
Oh no, I just wanted to double check that we can to say that DNDi can offer some support in generating solubility data or any ADME data, especially if, for example, if you want to cross-check your generated data in the lab to cross-check to different protocols, maybe even thermodynamic solubility, fast-safe or something like that. Maybe we can generate to cross-check or to generate for new compounds anyway. So just let me know.
Great.
We can do that for a handful of compounds.
Yeah, perfect. Thank you very much.
Yeah.
I mean, that's really useful for molecules which appear to be looking promising.
Yeah,
It's
exactly.
really
Yeah.
important to do the solubility thing before we get too excited.
Mhm.
I found a confirmed melting point, but it's from like a seller of this compound, so it would be like their quote. But it says that it's accurate. It says that it's not predicted. The melting point is 247 C for a related compound called trichlorosalicylanolide. So these may have some solubility issues. I'm not sure. They're very unusual though. These compounds have been around for a while, but I'm guessing the solubility is probably what stops them from getting in the clinic.
Yeah, possibly. Yeah. I mean, the absolute value gives you a sense of what it's like to be like. What you need is for a series, if you've got one with a high melting point and a lower melting point, that's probably tells you something about the relative solubility, but it's difficult to be sure about the exact amounts. But it gives you that number gives you a sense of low solubility. Yeah, Shane.
Yeah,
it was just
it lists the trichlorosalicylaniline as virtually insoluble in water, but soluble in alcohols and ketones.
All right. Okay. Yeah, Shane.
Yeah, I mean, another interest that brought me to this series was that I actually worked on a very similar series over 5 years ago, but this was to target some other sort of receptors, P2X7, and we used that same thought you had about using adamantane. And so basically it's almost like a flipped version, so the amide bonds swapped, and that methoxy and chloro group are also sort of swapped on that benzene ring as well. So, but we did a fair bit of data on that and some sort of pharmacokinetic data on that. So, very, very similar. We could sort of go off that sort of data as well, to sort of see what that would go like, particularly if you're thinking of substituting for the adamantane.
Yeah, look, there's a menu of possible isosteres that we could go for if we are excited and happy about the compounds, no problem, which should help with some of these issues. Yeah.
Maybe due to the sake of time, it's also good to look at the novel results Dunja created to aid in the discussion. Not created, tested, sorry.
Yeah, I don't know if I shared it, but I put it in the master list yesterday. I also tested the compounds on 2 micromolar in vitro against MM55. And I put it in a slide. I can share that real quick.
Oh, yeah, great. I should probably have started with this. Sorry.
Oh, I'm sorry. This went everywhere. My mind wanders.
So 6 of them have known structures. I don't know why there are no known structures for the last 4. They're not in the master list, the smalls. I don't know if someone knows it yet.
I just
But
noticed that as well, but I'll cross-check the Excel that Dr. Melander sent.
yeah, but these are the 10 that are very potent in vitro. I'm gonna determine the MICs hopefully before the end of this year. But I'm not a chemist. I don't know what this tells me, so that's what I wanted to show.
Just to clarify the numbers. So when you say minus 22.1% for the first compound.
Yeah, so so when you look in vitro, when you look at your plate, they seem to inhibit as much as itraconazole, but when when your APA measures the color, the balls are a bit lighter than your your negative control, which is itraconazole with the fungus.
So negative control will be adding just nothing, and that will be 100%.
No, negative control is the fungus with known antifungal, so itraconazole.
So what we noted from the past was that if we take a simple negative control, the fungus does something with the culture media as well, which changes the, even if it's dead. So we decided to do a negative control. You add the inoculum with a very high concentration of an antifungal agent, so that you know that the fungus is dead, so that any residual hyphae there would be similar to the ones which don't grow, because it did influence a little bit the reaction of the viability dye. So in that way, you get a better negative control. If you get a negative value, that simply means that the viability dye which we use is even lower than that of the negative control.
Right.
So negative values you can also read as 0.
I see. Okay. Wow. That's interesting.
Yeah, these compounds are, as far as I can tell, they're relatively potent.
Yes, it's also because I received, I think, 34 compounds, 33 compounds, and 10 of them are already very potent at the ultra low concentration. So that's something I noticed as well. I don't know how they would do in vivo, no time to test, but it's a good series to continue.
And I know sounds dumb, but if you leave out the hyphae, right, and you just have the molecule in vehicle, do you see any kind of anything? Because if the molecule is precipitating because of low solubility, is it interfering with the assay in a way that's giving you these numbers?
I haven't noticed anything like that, but that's also because we administer such a low concentration. And if we do the 100 micromolar, oh, I don't have the numbers here, but those are also not negative usually. So you would, if they would precipitate in medium, you would also notice that at a very high concentration.
Okay. All right.
Yeah, I
But you
mean,
don't typically run a control where you don't have any pathogen.
Yeah.
Right, so
Normally we don't do that because it's too much work to do that. So far, what we have tested never interfered with the viability dye itself. So if there's some interference with the viability dye, usually you see a change of color and not in the normal sense. So it changes instead of brown or orange to dark orange. Sometimes you see it changing to a completely different color, and then you know that there might be some chemical reaction. But yeah, it's not something we expect here.
Yeah, it could turn purple or like red, but I haven't seen it. But that also results in a very high number on the APOC reader. So you would have like 200, 300.
Yeah, so for those,
in the minus.
APOC is our spectrophotometer brand. Okay. So we measure this with the spectrophotometer.
Okay. All right. All right. I mean, if you're able to take one of the compounds which you have a lot of compound for and just check that the compound on its own doesn't give you a reading, it would. Yeah. I'm just worried about crystallization and precipitation or aggregation or something like that giving a crazy reading. But you know, I'm particularly with this series because the solubility is predicted to be solar. But yeah.
Yeah, I can do a quick test with that.
Yeah, I mean, not all of them. Just pick one that's performed well and just check it doesn't give you a crazy reading in the absence of the.
Yeah, in addition, you can actually buy very large quantities of the parent molecule, which is 5-chlorosalicylanilide. So that might not be a bad stand-in to do like phys chem testing on.
Right.
Because you can buy 5 grams for $30.
Okay.
On those ones which were missing structures, they're benzimidazole kind of derivatives. I've put a couple of them in the chat there. And that's why I didn't put them in the SAR diagram because I wasn't sure if we wanted them on the same page, basically. But I can probably pop them in there sometime tomorrow.
Fantastic. All right. Thank you very much. Look, we're coming up on the hour. So for this series, which has lots of interesting compounds in it that are looking promising, for this series, could people who are involved in it maybe just try and add a couple of sentences to the landing page, which is the repository homepage where the README is, and the wiki where we're talking about where this has come from, just to clarify for people what is going on here, because I think we've rushed into the promising biological data, but we haven't included where this is going. Sorry, where this has come from. So that's obviously, you know, partly also Bernard's story and
yeah,
how this has happened and where we are and who's involved. So just a few sentences would be helpful, just to clarify some of the history here to make sure we don't forget anybody. Would be useful. All right, we are running out of time, I'm afraid. I was just going to quickly mention that I submitted application to the Novo Nordisk antifungal call recently, and it's stage 1 of that call, trying to validate new targets for antifungal drug discovery where mycetoma is included as one of the relevant pathogens. And Wendy was on that, and others were on that. And we'll find out in December if we've gotten through to stage 2. But that would provide some resource to go after new targets. And I'm sure there's other things here, Wendy.
Yeah, I just put in the chat another option, which a colleague of mine sent to me. It's another grant application option for European partners. Non-European partners are also allowed, but I'm not sure if they will get funding. Where the aim is to find novel antimicrobial/antifungal agents, which could help the activity of the already known antifungal agents to reduce the risk of resistance. So maybe that's also an opportunity here, because the fungi listed on the fungal priority list are all allowed. So Madurella, mycetomatis, or the other eumycetoma causative agents would also be allowed.
Okay. So that you mean that would be what would be relevant is a target which helps reduce or make resistance less likely in combination with an existing antifungal? Is that what you mean?
So the call is called Identify and Develop New Combination Treatment Using Existing or Innovative Antimicrobials or Antimicrobial with Adjunctive Treatments to Extend Drug Efficacy and Combat Resistance. Right. So yeah, warring in bodies and the integrity pathway, all that stuff's relevant, right? Yeah, and proposals addressing this topic should identify and develop therapies to be used in combination to reduce the development of resistance against antibacterial and antifungal treatments and extend the usability of inexpensive and readily available antimicrobials. Right. These studies should be underpinned by a scientific rationale and mechanism of actions of these treatments. And in the framework of this topic, improvements of existing combination treatments is eligible. The choice of a targeted pathogen should be well justified. For proposals having a human health interest, the proposed combination treatment should be directed against one of the bacterial or fungal pathogens included in the WHO priority lists. Okay, so it might be an interesting option as well. Yeah, nice spot. Okay, great. Thank you for that. All right, we're on the hour. Any other really urgent stuff, or because we should probably finish up? Thanks for your time, everybody. Nice to see everybody.
Yeah, it's nice to see everyone. Definitely very interesting cell-analyte stuff. Definitely went down a rabbit hole with that. Very interesting. But thank you.
All right, see you next time. Bye.
Thank you. Bye-bye.
All right.